What is superbug? Superbugs is a term used to describe strains of bacteria that are resistant to majority of antibiotics commonly used today. But do you know, we are the creator of superbugs? Overuse and misuse of antibiotics help to create drug-resistant bacteria.
First, we should know why a bacterium can become a superbug. The first cause is mutation. Actually, mutation is a common phenotype in nature, such as in the remote past. Giraffes have different length necks resulted from mutation. In favorable environments, all giraffes with different length necks can eat leaves. However, when environments become bad, only giraffes with long necks can eat leaves. The giraffes owing shorter necks died of starvation. So in last, there are only giraffes with long necks on Earth. This phenomenon can also happen in bacteria. In some cases, the mutation made the bacteria resistant to antibiotics. So when treated with drugs, most normal bacteria died. However, the mutated one survived. Then the mutated bacteria can make the normal bacteria mutated. In last, they are all mutated bacteria which cannot be killed by antibiotics. When the mutated one can make the normal one having the ability to resist to drugs, this is resulted from the conjugation. When the bacteria is mutated, it owns a plasmid with a resistant region. Then it can contact the normal bacterium and replicate the plasmid with resistant region into the normal one, so the normal bacterium also gets the resistant gene. In general, mutation and conjugation work together, leading to the superbug existing.
What kinds of superbugs are we talking about? There are many types of multi-drug-resistant bacteria, but we are going to focus on methicillin-resistant Staphylococcus aureus, or MRSA, as an example. This drug-resistant bacteria can cause infections in many parts of the body and is a problem in crowded places such as hospitals. MRSA infection is caused by the bacteria Staphylococcus aureus. Staph bacteria are usually sensitive to beta-lactam antibiotics such as methicillin. These antibiotics kill staph bacteria by preventing them from synthesizing their cell walls. However, sometimes the antibiotics do not kill the bacteria. In this case, the bacteria has become resistant to methicillin as well as other antibiotics. It is now a superbug. Why is MRSA resistant to antibiotics? Through horizontal gene transfer, the staph genome acquires an extra gene called MEK-A. MEK-A makes a protein that helps the staph bacteria make their cell walls. This protein is not inhibited by antibiotics, so when MEK-A is incorporated into the staph genome, the bacteria become antibiotic resistant. The MEK-A gene is contained within a piece of DNA called scc-MEK. scc-MEK also contains genes that help it integrate into the bacterial chromosome. There are different strains of MRSA infections. This is because there are different types of scc-MEK chromosomes. Depending on which of these scc-MEK pieces integrates into the bacterial genome, a different strain of MRSA infection will result.
We need to protect our body from such superbugs infection. How can we prevent? Bacteriophages are currently being used therapeutically to treat bacterial infection. It has many potential applications in human medicine. Bacteriophages are bacterial viruses that invade bacterial cells, and they are much more specific than antibacterial agents. Bacteriophages recognize bacterial cells specifically and inject their own genome into bacterial cells. Then, bacteriophages grow into bacterial cells by utilizing their component and produce lytic enzyme to kill bacterial cells. Or, we can treat specific lytic enzyme directly to kill bacterial cells. But there are some problems remaining to treat safely and properly. For example, about resistance, the target bacteria may rapidly develop resistance against bacteriophages. And lacking of public awareness are one of the problems. Maybe some of you are afraid of threatening phage in your body. Superbug is a big problem in the world, so we need to learn this problem as a scientist. Finally, please confirm these 4 points not to produce new superbug. Thank you.